Good evening, students. So myself, Dr. Shantanu Sharma. I'm your Master Bio Faculty Teacher. So today we are going to study about that Megasporogenesis and Megagametogenesis. Today we are going to discuss about Megasporogenesis, means formation of megaspore, how your megaspore produce inside a female body inside the flower, or you can say in gynosium. Okay, let's see. See, there is one cellular mass which we call nucellus. Okay, now in this nucellus differentiation will occur. Okay, a type of division occur. Okay, and that type of division makes it to differentiate into two types of cell. Firstly, it will divide into your differentiate into archaeogonial cell. Okay, now this archaeogonial cell differentiate into two different cells. First one is your megaporogenous cell. Okay, now this, and second one is your megaparietal cell. Okay, now this megaparietal cell give its nutrition to megasporogenous cell. Okay, and after that megasporogenous cell converts into your MMC, megaspore mother cell. Megaspore mother cell. Okay, now you have a 2N structure which is your megaspore mother cell, and all these structures are 2N. Okay, all these structures are 2N in ploidy. Okay, now the previous megaspore mother cell will now go into a phase of cell division which we call meiosis. Megaspore mother cell will go in a process of cell division, meiosis, and go into meiosis 1. Meiosis 1. Okay, by meiosis 1 you have two megaspores. Okay, these are the two megaspores which is produced by the meiosis 1 process which is happening on your MMC. Now then again meiosis 2 happens. Meiosis 2 happens. And now these two cells again divide and produces 4 haploid cells. This is your 2N, this is N, this is N, this is N, 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 N. Okay, 4 haploid microspore develop after meiosis 2. Now see what happened. These 4 not going to be developed into functional megaspore. The one will be go and develop into your megaspore, okay, functional megaspore, and that one is this. The megaspore which is present on the chalazal end will differentiate, will convert into functional megaspore, while these remaining 3 will give its all nutrition to this one and die. ये खत्म हो जाएगा, this will be die, this will going to be die, and at last only this one remain. Okay, and this is your functional megaspore. This is your functional megaspore. Okay, now this functional megaspore now go into the second process which is called megagametogenesis. Means now we produce megagamete, female gamete, embryo sac. Okay, and this thing is present on the chalazal end of ovule. Okay, this thing is present on the chalazal end. Okay, and you can easily say the megaspore which is present on the chalazal end of ovule, it will differentiate, it will be conformed, it will be formed into functional megaspore. Okay, now next we are going to with megagametogenesis. Okay, now this N functional megaspore I am writing in short form. This is your functional megaspore. Okay, now this functional megaspore will go into first karyokinesis. Okay, 1K means first karyokinesis. Again, I am saying that this 1K is first karyokinesis, and this FMS is your functional megaspore. Okay, now it will go in first karyokinesis, and your nucleus which is present singly inside that your functional megaspore, it will differentiate, and you get 2 nucleuses inside a cell. Okay, now karyokinesis 2 happens. Okay, now these 2 nucleuses will divide and form 4. Okay, now karyokinesis 3 will happen, and 4 nuclei here and 4 nuclei here. Okay, one is your chalazal end, one is on your micropyral end. Okay, 4 on chalaza end, 4 on micropyral end. Now next is your migration. One nuclei from chalaza end and one nuclei from your micropylar end will migrate to the center of the cell. Okay, see, there is 4 nuclei, there is 4 nuclei. Okay, one nuclei from here, one nuclei from here, migrate in the center of the cell. And now next step is your cytokinesis, means formation of cells around the cell membrane, around all the nucleus. So three nucleuses are here. So 1, 2, 3. Okay, 3 is in the your micropylar end. 1, 2, 3. Okay, and 2 in the center. We'll draw this thing with different color. See that. 3 is here on your chalaza land. Okay, so this is your fully developed functional embryo sac or female gametophyte. Female gametophyte. Okay, now we have to give the names to these different cells. Okay, so this blue one on the top, your chalaza land side, is your antipodal cells. These two red one is your synergids. And this blue one is your egg cell, and combinedly they are called egg apparatus. And in the center, the big one is your central cell with 2 polar nuclei. So this is that upper one, that is the chalaza land, is your antipodal cell. The middle one is your central cell with 2 polar nuclei. Okay, and the lower one, these two are your synergids, and this one is your egg cell. And together they are called egg apparatus. Ploidy and N plus N. Point to be noted that this N plus N is not 2N. Okay, it is N plus N, just like a dikaryon stage we studied in fungus. Okay, this is N and this is N. Okay, that's why it is N plus N, not 2N. Okay, now this egg again N and this synergids again N. Okay, now there is some finger-like projections also present here in your synergids. Okay, some finger-like projections are also present in your synergids, which is called filiform apparatus. This thing is your filiform apparatus. This filiform apparatus is very important for pollen-piston interaction. These filiform apparatus secrete some type of chemicals which may attract pollen tube. Okay, the chemotactic movement of pollen tube, it is because of these filiform apparatus. They secrete different chemicals which attracts your pollen tube, and by pollen tube will come here and enter these their nuclei or that present a male gametophyte inside the synergids. Okay, so this is your fully furnished embryo sac, or we can say it in common way, female gametophyte. Mature female gametophyte. Okay, it is fully developed mature female gametophyte, and this full process is called Megagametogenesis. Okay, and this is the fully final structure of your embryo sac, which is ready for double fertilization. Okay, next video we are going to discuss about double fertilization. For that, for next time, like, share, subscribe, और कभी झुकने का नहीं.